Biomarker has pioneered the development of genetic technologies and industry since 2009 and owns more than 60 invention patents and over 200 software copyrights. Additionally, Biomarker has contributed to over 1,000 scientific programs published in high-level academic journals with a cumulative impact factor of more than 4,500. This continuous innovation in scientific research fuels Biomarker to become one of the premier promoters of genetic technologies in the era of biotechnology. Biomarker aspires to simplify gene decoding and accelerate advances in scientific research and is committed to provide standardized, customized and comprehensive technical services and solutions to customers all over the world. We have established 4 business segments devoted to high-throughput sequencing, bioinformatics, BioCloud, mass spectrometry, and intelligent manufacturing. In the field of next-generation sequencing, Biomarker leads functional genomics with SLAP-seq. SLAP-seq has accurately identified hundreds of vital functional genes that closely relate to human lives and has served a role in over 30,000 projects. Since third-generation sequencing came onto the stage, Biomarker became one of the earliest players in this new arena. We are proud to have introduced the PacBio platform-based tandem full-length 16S, 18S, ITS, amplicon sequencing technology and have launched Nanopore platform-based full-length transcriptome sequencing services, which brings microbiome and transcriptome study to a new level. The internet era has provided great opportunities in genetic technologies. Biomarker has efficiently and professionally integrated multi-omics-based big data and an online interactive analysis platform and high-performance cloud computers on BMKCloud, a self-developed online biocloud system. The platform is designed to help users interpret biodata and promote scientific research and health management, leading the generation of cloud-based genomics and biodata. Guided by the core values of continuous innovation and the pursuit of excellence, Biomarker provides its customers with high-quality services and products. At the same time, Biomarker has built state-of-the-art and automatic professional molecular experiment platforms, automatic and intelligent bioinformatic analysis platforms, and an information-based and standardized system to manage production, sales, staff, and finance. Biomarker will continue to create greater value for its customers and lead the industrial transformation with high-quality innovations to realize its dream of benefiting humanity with genetic technology. Biomarker Technologies. Innovation leads to wonders. Biomarker has pioneered the development of genetic technologies and industry since 2009 and owns more than 60 invention patents and over 200 software copyrights. Additionally, Biomarker has contributed to over 1,000 scientific programs published in high-level academic journals with a cumulative impact factor of more than 4,500. This continuous innovation in scientific research fuels Biomarker to become one of the premier promoters of genetic technologies in the era of biotechnology. Biomarker aspires to simplify gene decoding and accelerate advances in scientific research and is committed to provide standardized, customized and comprehensive technical services and solutions to customers all over the world. We have established 4 business segments devoted to high-throughput sequencing, bioinformatics, BioCloud, mass spectrometry, and intelligent manufacturing. In the field of next-generation sequencing, Biomarker leads functional genomics with SLAP-seq. SLAP-seq has accurately identified hundreds of vital functional genes that closely relate to human lives and has served a role in over 30,000 projects. Since third-generation sequencing came onto the stage, biomarker became one of the earliest players in this new arena. We are proud to have introduced the PacBio platform-based tandem full-length 16S, 18S, ITS amplicon sequencing technology, and have launched Nanopore platform-based full-length transcriptome sequencing services, which brings microbiome and transcriptome study to a new level. The internet era has provided great opportunities in genetic technologies. Biomarker has efficiently and professionally integrated multi-omics-based big data and an online interactive analysis platform and high-performance cloud computers on BMKCloud, a self-developed online biocloud system. The platform is designed to help users interpret biodata and promote scientific research and health management, leading the generation of cloud-based genomics and biodata, guided by the core values of continuous innovation and the pursuit of excellence. Biomarker provides its customers with high-quality services and products. At the same time, Biomarker has built state-of-the-art and automatic professional molecular experiment platforms, automatic and intelligent bioinformatic analysis platforms, and an information-based and standardized system to manage production, sales, staff, and finance. Biomarker will continue to create greater value for its customers and lead the industrial transformation with high-quality innovations to realize its dream of benefiting humanity with genetic technology. Biomarker Technologies. Innovation leads to wonders.
Good morning or afternoon. This is Lydia from BMKGen. I'm thrilled to welcome each one of you to our latest webinar. Today, we will explore analysis real data with BMKCloud in the case of mRNA-seq. Our webinar today will last for about 40 minutes, followed by a dedicated 5 minutes for your questions and answers. You can freely submit your questions by using the Q&A button at the bottom of your screen. We're looking forward to receiving your inquiries, and if any questions are left unanswered, we will contact you to reply via email. Also, we'll share the slides and a recording of this webinar via email. In this webinar, we will explore the application of mRNA sequencing on BMKCloud, a comprehensive bioinformatics platform. The following topics will be covered. Firstly, introduction to material and treatment of BMKCloud. Explain treatment and group setting in the case of plant material and mango setting. Secondly, basic analysis result. We will talk about this from 3 parts: analysis submission, report check and update, obtain basic images, and intersected target gene list. There are demonstrations followed. And thirdly, advanced analysis, WGCNA submission, introduction, reason, result, explanation, Venn diagram, and image customize. But before we start, let me introduce our distinguished speaker for today's webinar, Johnson, equipped with extensive expertise in bioinformatics analysis, shines in his role. Holding a master's degree from Beijing Forestry University, China, he embarked on his professional journey as a bioinformatics analyst post-graduation, specializing in hexapod sequencing data analysis, especially Pacific Biosciences data. Johnson honed his skills with precision and dedication. His career trajectory led him to BMKGen, where he transitioned to the overseas bioinformatics department. Here, he assumed the pivotal role of cloud platform product manager, steering the team towards innovation and efficiency. With a blend of technical prowess and strategic vision, Johnson continues to make significant contributions to the field, driving advancements in bioinformatics with unwavering determination. Without any delay, let me hand over this virtual stage to our master, Johnson.
Thank you, Lydia. Hi, it's Johnson. Today, we will talk about analyzing real data from mRNA-seq reference in BMKCloud. First of all, let's review what we've talked last webinar. You should know that BMKCloud can offer a one-stop solution. We have bioinformatics tools such as image generator, sequencing analysis tools, and data processor. You could use online analysis applications to do some basic data analysis. BMKCloud provides various analysis applications. After you get your final report, data mining or customize, you can do this without any limitation. You can change colors, reset groups, or other customized settings by yourself. We talked about from real data to a report, such as submit task, monitor analysis, update report, and download the result. We can get expression matrix and basic visualization results. Next, we talked about in-depth result filtering. For example, select gene by feature annotation, functional annotation, or motif. We can get target gene list. And we talked about how to from gene to visualization results. We did further image adjust, such as font family, axis title, or boundary size. We can get some further visualization results. For today's presentation, we have 5 topics. Number 1, I will introduce the material and the treatment of the real data, and how to do the basic analysis from data to report. And I will use WGCNA as an example. I will introduce the principle, bioinformatics principle, and the results. And I will use the Venn diagram as an example for image customize. And last, I will summary for today's webinar. So let's turn to the material and method. Today, we will analyze mRNA-seq reference with real data. First, let's see the material. All analyses were grown on MS media in short stakes condition at 20 degrees for 21 days. Cold treatments were performed at 4 degrees in short stake condition for 3 days or 3 hours, and 3 seeds treated as naive. So we have 3 groups, so each group has 3 repeats. Here I cite the differential expression analysis compare groups, naive versus H3, naive versus D3, H3 versus D3. Our main goal is to analyze the impact of cold stress on plants by examining gene expression. For this part, today we will follow these steps. Number 1, conduct analysis and check data quality. Number 2, review grouping condition and assess whether analysis parameters need adjustment. Number 3, summarize the current analysis results. And number 4, conduct advanced analysis and refine conclusion. Next, I want to check mapping. I have a pre-prepared analysis report, and here is the result. The first step, I want to check mapping ratio. Here, the mapping ratio are over 95%, and more than 90% for unique mapping ratio. So I think the quality of my data is okay. Further analysis is meaningful. Otherwise, we should deal with quality of data firstly. Next is the group sightings. To this step, I think my data quality is okay for further analysis. Now I wonder, whether my group sightings are suitable for my data. Here, I check the correlation and the PCA results in my report. I think my H3R1 sample and the D3R1 sample are out of groups, right? Both of these two images show this. I consider to remove these two samples in order to get more reliable results. Then I check parameter sightings. Here, parameter sightings meaning differential expression analysis parameters. Now, I set FDR to 0.01 and fold change to 2. I have 3 compare groups. First, let's check total differential expression genes number. There is 1,000 to 2,000 DEGs. I think this is an acceptable level. Furthermore, group naive versus treatment group, including naive versus H3 and naive versus D3. They have similar differential expression genes number. This suits my experimental design well. We checked my analysis report by far. I checked the data quality, mapping ratio, DEG analysis parameter setting. After all this checked, I decided to remove H3R1 sample and D3R1 sample and keep DEG analysis parameters. Do you remember how to update report using BMKCloud? Let's do this together. Here is the main interface of BMKCloud after you log into this website. And once we want to update the final report, the first step, we must opening the previous report. Here is my report I just prepared. And you'll remember on the left, this is the main interface of the final report. And on the right is the catalog. On the top, you can download the final report, right? And on the left is the navigation bar. Once we want to update the final report, so click here, update the final report, samples and group sightings. Remember that we just talked about I want to remove 2 samples, H3R1 and D3R1. So I click all samples and unclick D3R1 sample and H3R1 sample. So I finished the BMKCloud will automatically finish the analysis without these 2 samples. I want to keep the differential expression analysis parameters. So this part for the FDR and fold change won't be changed, won't be adjusted. For the group sightings, I have 3 groups and 3 compare parts. So because I just removed 2 samples, so I have to reciting the groups. I have 3 groups. The first is the naive group, containing the 3 samples, naive 1, naive 2, and naive 3. And I have 2 treatment groups, such as D3, including D3 repeat 2 and D3 repeat 3. Another treatment group is H3, including the H3 repeat 2 and H3 repeat 3. So I have 9 samples from the very beginning, and now I have 7 samples in the updated report. For the compare groups, I have 3 samples. I have 3 groups, like naive versus H3, naive versus D3, and H3 versus U3. After change all these parameters, you can set your new report a new name so you can find or you can recognize what we've done. Just for me, I want to title it with remove H3 R1 B3 R1, and this data is after you change all these parameters and you can submit the analysis. We talked that our group updating will take less time than the first analysis because we don't need to realign the reads to the reference genome. After you submit analysis, you can find this task at here, project task. Here you can see the analysis we just submit. It is contain running, has the label running, which means our analysis is automatically running on the BMKCloud. Once the analysis is finished, you can check this label is turned to complete, which means you can open the final report. So this is how to update the final report. I remove 2 samples and keep the parameters and group settings. Let's back to my slide. We removed 2 samples and retained the original parameters for differential expression analysis. Now, let's set the obtained results. Because of the time fluctuation and the time cost, I have a pre-prepared result here. So let's check the result we just obtained. First of all, after removing the samples, the PCA analysis here and the correlation analysis result here are more in line with our expression. Although the naive group is still a bit scattered, I think this is acceptable as the control group here. The number of DEGs has reached a similar level here. And the data account is not too large, which I found acceptable. According to the Venn diagram here, the number of intersecting genes is still considerable and can be used for further analysis. After updating the final report, we've got target gene list for differential expression genes, or gene list of target functional annotation. We've got basic visualization results, including functional enrichment and gene expression cluster analysis. For the advanced analysis tools, I will use Venn diagram and WGCNA as an example. The first part, I will demonstrate you about Venn diagram. First, we will talk about how to get a Venn diagram using BMKCloud. After we get the plot, I will show you how to save results by file, so you can find this result anytime you need. Then you can regard Venn diagram as a tool for us getting intersected gene list and do further analysis. So let's turn to BMKCloud, and I will show you step by step. Let's back to the interface of analysis report, and I will use Venn diagram as an example. First, let's find the Venn diagram on the left. Here, gene expression, gene set Venn diagram. Click it. And you can change the parameters of the Venn diagram. This part, I will use whole gene list for the further analysis. Otherwise, you can use the differential expression genes for the further analysis. Here, do you see? On the top is the analysis based on the whole gene list. And here, you can use the differential expression genes for the further analysis. Such as this, I have two analysis parameters. And I use this one, and this one. After we select the gene site, I can submit analysis. Wait for a minute, please. The BMKCloud will automatically complete the analysis. If we have many differential expression genes, this may take a little bit long, but for the analysis, I don't have much more disease. This may take less time. So here you can see we got an analysis result for the total differential expression genes, and we got the Venn diagram. You can customize this Venn diagram by clicking this image. Click it, so you can see there's all these customized tools is listed here. You can just analyze the text for the family, for the size, or the color size. Maybe you don't like this color, you want to change it for the NPG. So you can just click it, and this will automatically change it. For the layer setting, you can change whether to hide the legend. You see all the personalities, such as group boundary, all this. See. Here I want to see about that. I want to say that we maybe I just submit analysis and do the customized tools, but I must quit now, have a break, or we have other things to do. So I can save the current analysis result into BMKCloud. So you don't have to download the middle files such as the table or the middle results such as the image into your personal computer. You can just save that into the BMKCloud. First, we save the current picture to the personal data, so the file name you can change. And okay, this image will save to this diary, so you can find the SVG file for this Venn diagram. And besides on this, I want to save the gene list, this analysis workflow. What should I do? Here, we have two samples. The first one, we can save the gene list. So I can just here, click the save, list save. This will save the gene list of this table into the BMKCloud, or you can just download the gene list here. A new window will be opened and the list of these genes will be downloaded into your personal computer. On the other hand, you can save the analysis results here, which means we can save all these parameters, all these tables, all these images by file into BMKCloud. Just as I said, you don't have to download all this. After you can break here and save this result. Maybe I want to name the and click OK. All these middle results will save into BMKCloud. And where can we find the results here? By file. Here, you can click the process record. You can guide this finished Venn map and today's data. Click it. We will open a window. This is the same page as this. So as the Venn diagram submission page we just used, you can get the very same results by far. So you don't have to download all these files or the middle results to your personal computer. I think it's very convenient for the analysis. So this is the Venn diagram. Let's back to my slide. Let's summarize here. We talked about how to submit the Venn diagram step by step. using not only all genes, but also DEGs. We can save and find previous results using BMKCloud, saving temporary results into process record. We can intersect gene using BMKCloud and using these genes for the further analysis. Oh, I forgot this part. Let's check. Let me show you this part in BMKCloud. Here is the result of Venn diagram after we clicked in this image. You will find that all these numbers is clickable, which means if you want to use this part genes like this about 700 genes, you can just click this number, or you want this, or you don't want this. You only want this intersected gene for the 3 groups. You just click that number. Then you can find here. Data filtering is clickable. Click it, you will get the gene list for the intersected gene for the about 700 genes. Then you can get the gene list. You can download or use this gene for the further analysis, such as one-click drawing and this. It's very convenient for using BMKCloud to do all this analysis. Now let's turn to WGCNA. Maybe you've already heard of this method, but don't know about the details. Let me introduce to you simply. The full name of WGCNA is Weighted Gene Co-expression Network Analysis. WGCNA is a technique that looks at the relationship between different genes in a large data set, typically genes expression data. The goal is to identify groups of genes that behave similar across samples. These groups or modules may share common function or be involved in same biological process. So why we will use WGCNA? There are 3 reasons. WGCNA can identify gene modules, and WGCNA can link modules to trees, and WGCNA can do the data reduction. Using these advantages, we will use WGCNA for mRNA-seq analysis. Here are the main steps of WGCNA, including data preparation, network construction, module detection, relating modules to trees, and module analysis. The in-depth understanding for this need not only biological knowledge, but also mathematics background. For most of us, we only need to know that WGCNA helps us find many genes related to few genes and find few genes related to many genes. Next, I will demonstrate to you about how to submit WGCNA using BMKCloud and go through the main results. Let me introduce parameters of WGCNA in BMKCloud for you first. We should choose about genes we use for analysis. We can select DEGs in specific compare groups. If you have many samples and groups, you can just merge samples, which will decrease complex of analysis. Otherwise, you can use all your samples. Notice that WGCNA performed well using at least 5 samples or groups. Next part is gene filtering. You can filter genes using expression, such as filter out genes FPKM less than 1. For WGCNA module filtering, if you don't know how to choose these parameters, leave this by default. You can also add traits acceptable. If you have quality traits, you should compare these traits into integers such as 0 and 1. Talk is cheap. I will demonstrate to you WGCNA using BMKCloud. Let's turn to BMKCloud. Here, Let's back to the interface of the final report, so you will think it's very familiar for you. Here is the result of this is the main interface of the final report, and I want to use WGCNA for the further analysis. I will use the DEGs for the further analysis. So click the differential expression GIS analysis, and here you can find WGCNA. Click it. Here is the main interface for the WGCNA analysis submission. And the first step, did you remember? We must choose the gene set for the further analysis. Click here. I want to use for these parameters for the FDR for 0.05 and for the change for 2. For the samples, because I only have 9 samples, I will not merge samples into groups. If you have many samples, you can click here and you can merge the samples into groups for the further analysis. I have 9 samples, so I clear all these samples. For this analysis parameters, if you don't know how to set this or how to filter the genes, you can use the default parameters we recommend, and you can change this parameter after you got the analysis result first. And do you have the treatise such as weather or the other? If you have the treatise such as the weather or the flowering or the whites such as this, you can use this. Click yes, and you can set the trees of all these samples. I will write 2 examples. The first one is the quantity trees, such as the weight. The weight of the 9 samples, maybe I will type into this assembly. For the quantity trees, you can use this integers into the analysis. And I will add the trees for the quality trees, such as flower or not. For the flower or not, this is quality. It is a quality trace, so I must convert the quality trace into the quantity trace, such as using the integer 0 and 1. You know what I'm meaning, right? After you set all these parameters and trace, you can submit analysis. BMKCloud will automatically finish the analysis. And you can check the result using this process record here. Let's back to my slide. If we WGCNA analysis will take a little bit longer time. So I have prepared the result, and let's go through about the main result of this. First table you should focus on is the gene list with modules. WGCNA will divide genes into modules and use different colors respond for modules. In this table, we can know that some genes, such as this, is in blue module. Here, we should download this list for or save this result in BMKCloud. Next, we should focus on the result of module-trait relationship. In my result, we can find that blue module significantly correlation with the flower trait. Here, this part, and brown module here, and brown module significantly navigated negatively with correlation with the height trait here. If we want to analyze further with these 2 traits, maybe these 2 modules needs our more attentions. I want to know how code treatment influence, so blue module and brown module fit my design well. We can find all the gene modules relationship and functional enrichment results in download zip file. But the results here are for a specific module. I want to enrichment results for both two modules. What should I do? For functional enrichment analysis, I want to show you how to use combined gene list for further analysis. I analyze the code treatment with two trees, flower and height. I want to go further about influence details, so I have two ways based on current results. First way is getting common gene set for target trees and DEDs. After getting gene set, using functional enrichment result for check, maybe we already have a large gene set for all DEDs, and a smaller one for gene set related to treat height, and another gene set which is treated with treated flower. So we can get a smaller gene set with intersecting gene for all these gene sets, and using functional enrichment result to check whether the pathway related to cold stress. Then you can select genes for expression gene design. The second way is collecting a common gene set for all genes, which we are interested in. Then using functional enrichment or further selection to get your final gene list. The difficulty of this approach is how to get a gene list based on functional enrichment results. But in BMKCloud, you can just combine two gene sets simply without distinct genes. I think this is very simple. So we talked about BMKCloud, updating final report, WGCNA, Venn diagram, and enrichment function result. That's all. Thank you. Any questions?
Okay, thanks for Johnson's presentation. He explains the topic in great detail. I believe you have a good understanding of analysis real data with BMKCloud in the case of mRNA-seq. Now you can submit your questions through the Q&A button at the bottom of your screen. We received some questions regarding the topic. We will answer two of them on set. The rest will be answered by email. The first question, for Venn diagram, can any part of the diagram be selected, or just the intersect part?
Oh, thank you, Lydia. Each part of Venn diagram can be clicked. I will show you in this slide. You can not only select the intersect part, but also you can select both parts such as this and this, this and this. You can select any part of in Venn diagram for the gene filtering and do the further analysis, such as the one-click drawing for the GOKGG functional analysis and the COG enrichment analysis.
Okay, the second question is, does BMKCloud provide all results of WGCNA package or just the results you shared?
Thank you, Lydia. This is a good question. We just went through about the main results in WGCNA analysis, but you can see in this page that after you download the whole zip file for the WGCNA, you will get all these results like this, such as the enrichment and the heat map. Not only the results from you can not only get the whole result of the WGCNA on that WGCNA package, but also you can get the functional enrichment result from BMKCloud. This is very helpful for your further analysis.
Okay, thanks for Jonathan's answer. But due to time constraints, we apologize for not being able to answer all questions, but we will answer them by email after we have finished. So you can wait for our email. As we conclude today's session, I want to express our gratitude for your participation. Look out for the follow-up email with the webinar summary and the promised recording. And make sure you check our social media page for future webinars. Thank you again. Have a nice day.